Student, in the last video we studied the external morphological features and anatomical details of the pteridophyte Selaginella. Now we also want to learn the reproduction and life cycle of Selaginella. The plant we see in the nature, that is a sporophyte of Selaginella. That sporophyte is diploid in nature. This sporophyte grows prostrate or in a erect manner. Some of the vegetative branches from this sporophyte becomes a reproductive, and at the time of reproduction they produces the special kind of structure at the tip, and that structure is called as a cone or strobili. This cone or strobili take part in the sexual reproduction by producing the microspores and megaspore. That way, the Selaginella reproduces vegetatively and sexually. The vegetative reproduction in Selaginella is somewhat rare, that is not so common. The vegetative reproduction sometimes takes place by formation of tubers and bulbils or by formation of fragmentation. The sporophyte get under fragmentation, or they may form a tuber or bulbils, and these structures in the favorable condition germinate and forms a new sporophyte. This is what a mode of vegetative reproduction. The important kind of reproduction takes place in Selaginella, that is a sexual reproduction. As I said, the sexual reproduction takes place by formation of the male and female gametophyte. As you know, the Selaginella is a heterosporous plant, and at the time of reproduction, some of the branches of sporophyte grows vertically erect and forms a strobili or cone. That strobili or cone is also heterosporous, and that forms at the tip of the reproductive branches. You can see in the figure, at the tip of the branches you will see the cone or strobili. Sometimes the vegetative branch can grow extends and forms another cone in case of the some species. Otherwise, the single cone is produced at each tip of reproductive branch during the time of reproduction. The cone or strobili is a heterosporous in nature. This cone or strobili bears two kind of leaf-like structure, and that structures are called as a sporophylls. Sporophylls means the spore bearing leaf, and that's why it is called as a sporophyll. At the adaxial base of the leaf, the spore is present. There are two kinds of sporophylls are produced in the Selaginella, and they are respectively microsporophyll and megasporophyll. The microsporophyll bears a microsporangia, and megasporophylls bears a megasporangia. These two sporophylls are spirally arranged on the central axis of the cone or strobili, and they form a compact cone or strobulus. Generally, the megasporophylls are seen present at the base of the cone, and towards the upper side the microsporophylls are seen present. Sometimes these both sporophyll can be alternately present. At one side or one side there may be a microsporophyll, and another side there may be a megasporophyll, like that. But in general, megasporophylls are seen produced at the base of the cone, and microsporophylls are seen produced at the upper region of the cone. Each micro and megasporophyll are present inside the microphyll and megaphyll, means they are produced inside the microleaf and megaleaf of the cone, and they are spirally arranged. If you will see the LS of cone, you will see there is a megasporophyll. At the megasporophyll base there is a ligule, and inside or inner side of the megasporophyll there are megasporangia. You can see in the figure the lower portion showing the presence of megasporangia, and inside the megasporangia you will see the 3 megaspores are seen developed. Similarly, on the upper region you will see the microsporophyll, ligule and microsporangia. The microsporangia is also a sac-like structure, and inside this microsporangia you will see the number of microspores are produced. The cone or strobili has a central axis, and alongside of the central axis or around the central axis these micro and megasporophylls are spirally arranged and they develop a cone or strobili. This is what a structure for the formation of these spores in the Selaginella. So Selaginella is a heterosporous kind of plant. The cone of Selaginella is broad at the base and becomes narrow or tapering towards the end. Now we will see a structure of microsporangia and megasporangia. First we will see a structure of microsporangia. The microsporangia are produced at the inner side of microsporophyll. This microsporangium bears or produces a microspore. A microsporangium is ovoid, reniform or spherical in shape. It is smaller in size than the megasporangium. The microsporangium is a two-layered structure, and next to this two-layered there is a tapetum. Inside the microsporangium there are presence of microspore mother cell. This microspore mother cell undergoes the meiosis and they forms a large number of microspore, and these microspores are developed inside the microsporangia or a bag-like structure. The megaspores are produced in megasporangium. The megasporangium are located towards the inner side of megasporophyll at the base of the strobili or cone. The megasporangium has a outer megasporangium wall, and inside this wall there is a tapetum. Inside the tapetum there are megaspore mother cells. Large number of megaspore mother cells are present, but only one remain functional from this large number of spore mother cell, and another get degenerated. This functional one undergoes the meiotic divisions and forms a tetrad of 4 haploid spore. These tetrad of haploid spore are nothing but a megaspore. There are 4 megaspores are developed by each megasporangium. When these 4 megaspore are fully mature, that time the megasporangium has a only single layer of jacket. The megasporangium dehisces and these spores are get scattered and disperse at a great distance as compared to microspores. The structure of megaspore as compared to microspore is somewhat larger. These megaspore are present in the group of 4, and they shows a triradiate mark at their apex. The megaspore has a 2 wall. The outer one is exine and inner one is intine. The outer is cluttered and inner is uniform and intine. The microspores are smaller in size. They are pyramidal shape and have a thick ornamentation of ornamented exine and thin uniform in time. Both these spores have a nucleus and reach in cytoplasm. Later on, these two spores, megaspores and microspores, are the representative sexual reproductive structure for the formation of male and female gametophyte in the Selaginella. The microspore in the Selaginella develops the male gametophyte. The development of male gametophyte started within the microsporangia. In the development, microspores divides into two unequal cells. One is a larger and one is a smaller. The larger upper cell is called as antheridial cell and smaller cell is called as a prothelial cell. In the further development of the male gametophyte, the prothelial cell forms a covering around the antheridial cell, and antheridial cell forms a antherozoid mother cell or androcytes inside the antheridial cell. Large number of antheridial cells are produced. There are about 128 to 256 androcytes are produced by the antheridial mother cell. Each antherozoid or androcyte is a biflagellate structure, and at the maturity the jacket cell of the male gametophyte are get disintegrated and the antherozoids are liberated in the surrounding water. Whereas the megaspores also germinate in the megasporangium, and after germination the megaspores forms a female gametophyte of Selaginella. This female gametophyte has a archegonia, and at the time of fertilization the liberated antherozoids by swimming reaches to the mouth of archegonia. At the time of fertilization, the neck canal cell and ventral canal cell of the archegonia are disintegrate and they form a passage for the entry of antherozoids. With the help of that passage, antherozoids enter in the archegonia and fertilizes the egg. The product of fertilization is oospore or zygote that is again diploid in nature. This oospore or zygote get converted into the embryo itself in the megasporangium in the cone, and this zygote or embryo is a beginning of the sporophytic phase. Afterward, this zygote or embryo germinate itself in the cone and produces the young sporophyte of Selaginella. In this way, Selaginella reproduces sexually by forming male and female gametophyte and fertilization. At the end of the study of Selaginella life cycle, we will see the diagrammatic representation of life cycle of Selaginella, how the Selaginella completes their life cycle. The life cycle starts from the mature sporophyte, that is a mature plant we see in the nature. This mature plant at the time of reproduction produces the cone or strobili at the tip of fertile branches. This cone or strobili is a heterosporous. It shows the presence of microsporangia as well as megasporangia. The microsporangia is a representative of male gametophyte and megasporangia is a representative of female gametophyte. These two structure, microsporangia produces the microspore mother cell and microspores, whereas the megasporangium produces the megaspore mother cell and megaspore tetrad. These two spores, microspores and megaspores, latter germinate and respectively produces the microgametophyte and megagametophyte. The microgametophyte contains a antheridia, and that antheridia produces the biflagellate sperms or antherozoids, whereas the megagametophyte produces the archegonia, and that archegonia possesses the egg at their solan base or venter. During the fertilization, the liberated biflagellate antherozoids, you can see in the figure also, reaches to the egg and fertilization or syngamy takes place. The product of fertilization or syngamy is a formation of zygote. This diploid zygote undergoes the further development and it starts germinating and produces the young sporophyte. This young sporophyte in the latter stages of development get developed into the sporophyte of Selaginella. In this way, the Selaginella completes their life cycle. The life cycle shows the two phases, sporophytic phase and gametophytic phase. As compared to sporophytic phase, the gametophytic phase is of a very little time period, and that's why the sporophytic phase is seen dominant, and that's why the life cycle of Selaginella is diplohaplontic kind. During the formation of male and female gametophyte or microspore, meiosis takes place, and at the time of formation of zygote, syngamy or fertilization takes place. Here the two gametes are get fused and they form a zygote. This diploid zygote then later undergoes germination and develops a new sporophyte. So in this way the life cycle of Selaginella is get completed.